we know at best 25% of the species that exist right now in this planet. Good afternoon. My name is Manuel Caballero Gutierrez. I'm a Spanish biologist working in the exploration of the ocean and its diversity. I have the privilege of describing and naming new species. And believe me, there are many new species. Actually, there are millions of species to be described. But how many? To answer this question, we have to go back to the 70s. You know, ABBA, Rolling Stones, odd haircuts, nuclear weapon testing. And in that moment of history, in the 70s, we knew about 1.6 million species. And we thought that this number represented half of the species to be known. But then an entomologist called Erwin in 1982, studying 13 trees in the Costa Rican tropical forest, made an estimation of 30 million species of arthropods only in the tropical forests of Earth. And that changed our view about the proportions and the magnitude of biodiversity, of the number of species on Earth. That started a series of works, like a fever of the study of biodiversity on Earth, that gave us a range of estimations on the species on Earth that goes from 5 to 100 million. 1 trillion if we include bacteria. But don't worry about this. The most accepted estimate up to this date is of 8.75 million species on the planet. That means that we know at best 25% of the species that exist right now in this planet. Now, I am a Star Wars geek and a taxonomist, and probably all the normal people that is today here with us have no idea of what taxonomy means or what is formally describing a species. What is a species formally described? Well, a species is formally described when it's found. We will go back to that. It's studied anatomically or DNA or with other techniques, and it's named based on a horrible maddening book called the International Code for Zoological Nomenclature in my branch of science, and then published in a scientific journal. I promise you this is a very long and tedious process. To answer the other question about taxonomy, taxonomy is the science that study how species distinguish themselves, and we put that in an evolutionary context. How we can know from the morphology of the species or from other characters how the species are related to each other. That's a line between taxonomy and what is called systematics. But going back to taxonomy, one of the main purposes of taxonomy is to find and name and describe new species, of which, as I say, there are many. To recapitulate, we thought we knew most or half of the species on Earth. That was in the 70s. Now we know that we might know 25 at best. 15 to 20,000 species are described a year. With the accepted estimate, that will mean that we need around 500 years to describe all the species on Earth. I will be retired by then. I won't wait that much. But the important point here is that there are not sufficient. There's not a sufficiently big number of specialists. Actually, many specialists die while still working. There's a case of a Japanese guy that at 99 died, and a couple of years later still was publishing papers after his death. So that gives you an idea. The problem is that when we die, our knowledge dies with us. And believe me, we need to study, we need to identify and know new species, or the species that exist at all. But if you're not a specialist, you will ask, yeah, well, why do I care? What is biodiversity important? What is all this fuss about it? Well, the answer, my no-geek friends, no taxonomist friends, is that the ecosystems, biodiversity, sustains human life on Earth. We actually use the services provided by every ecosystem on this planet. Everything you ever ate, everything that you're going to eat, even this pencil that you ate when you were in first grade, comes from biodiversity. Yeah, it was mostly made of wood, right? All the oxygen that we breathe in the air comes from a group of bacteria, small called cyanobacteria, that around 2.5 billion years ago changed the proportions of gases in the atmosphere with a new process in that moment called photosynthesis. And that is what we breathe today. Every photosynthesizer in this planet provides the means for humans to be able to breathe. Every tissue that you have on you right now, cotton, wool, silk, even the synthetic tissues coming from petroleum come from biodiversity. Petroleum comes from biodiversity. All the materials that we use in construction, like wood, come from biodiversity. Biodiversity regulates climate, cleans the water that we drink, and even provides beautiful places to go and picnic. The aesthetic value of biodiversity, of the ecosystems, hasn't been really appreciated up to now. So please, don't save the planet. Do not save the planet. The planet doesn't need to be saved. We need the planet. We need a healthy planet with healthy, fully functional ecosystems to sustain human life on Earth. That is especially important in the context of global change, right? Because we are affecting the proportions of gases in the atmosphere, changing the amount of energy that is available, and therefore changing the weather patterns. And those changes are too quick, and they are causing what is called the sixth mass extinction. The first one in history, in registered history, that has been caused by humans. When a species faces changes, a big change, it has three options. Move, which is kind of difficult for a tree. Evolve, which doesn't happen in a generation. We don't have this kind of time right now. Or die. The species gets extinct. So, again, why do we need to preserve biodiversity? Well, we need to preserve it because we need to save ourselves. And how do you preserve and manage something? Well, you need to know it first, right? So how do we study biodiversity in the modern era? Well, we do it exactly as it was done 200 years ago, but more efficiently, and we use DNA and diving equipment, at least for the oceanographic studies. Every time that you hear a politician saying, we know biodiversity in the ocean because the satellite images, well, with satellite images, you can know the proportion of chlorophyll in the surface of the ocean. You can estimate the biomass of the forests. You can even estimate the biomass of phytoplankton in the ocean. But to know the number of species, the number of different species in the planet, you need to go there and pick them one by one. So traditionally, taxonomists have been loners. When I was starting, you were basically a guy that went to a tropical place, made his own samplings, processed the samples, went back home, did anatomical works, and described new species from foreign countries. this view has changed and had to change because there are so many species that this system is not working. One of the ways of improving that, of improving and accelerate the catalog of life, is the one used by research institutions, some research institutions that organize big, very big surveys, including many scientists in remote areas to study new species or species at all. I am going to use my experience with the National Natural History Museum in Paris, in which I worked for 7 years, to describe a workflow in a biodiversity exploration campaign in a tropical place, with a brief citing some of the images that are available for me. So basically, you have this vessel from which you make dredging, getting sediment with specimens. And you also have a team of divers that pick the samples one by one, or brush surfaces to take to the lab, or make vacuum sampling. Basically, you put into the ocean a vacuum cleaner or something like that. You can see it in the image. And you bring those samples to a temporary lab and you put them on seawater. You use a mesh of, or several meshes of different sizes to separate the samples, and the samples stay separated by size in ocean water. Then they are picked and sorted by specialists or collaborators, and then they are photographed alive. They are tissue clipped for being able to study DNA. They are labeled, and then they are preserved. Typically, one of these huge expeditions produces several cubic meters of minute samples, clean, labeled, that are taken back to Paris in this case, where they are stored, sorted by group or family, and where they wait for the roughly 200 specialists that work with us, well, I keep working with them, to name and describe the new species that may arise in those samples. The whole process of describing a new species usually takes 10 to 20 years, sometimes way more. In about 25 years of work, I have described 72 new species, 5 genus, and even 2 subfamilies. They have beautiful shapes, magnificent colors, and they are really small. Actually, this one is about 2 millimeters long. Those are species of Gibberulas from the Caribbean. And this comes from a mission with the Natural History Museum from Guadeloupe. It's called Ipsilodoris fregona because of how the gills are turned around themselves. I expect to double this number this year with my study of a family called Marginellidae from South Madagascar. But where all these questions arise in my being, right? Well, I was born there. I was born in this beach, not literally, but like maybe 1 kilometer away. I learned how to dive, how to swim. I saw my first mollusk. I learned how to sail, how to windsurf, how to surf. I did my PhD there. And all my questions come from how I was raised in that place. I'm being emotional right now. My father always says that he's really happy and proud of me being doing my PhD there. Well, that's a photo of me in this place when I was like 3. But it is not all a paradise. Actually, the life of a scientist working in biodiversity is really hard. Funds are scarce and jobs are actually rare, and you find your stability when you're around 40 years old. That makes really unlikely for you to form a family. By fortune, I found a person, another biodiversity expert, that wanted to have kids with me. And actually we have two. And you can see in this image our eldest working with us during COVID times in Corsica in an expedition organized by the Natural History Museum in Paris. They follow us wherever we go. Well, one of the things that scientists has to do is spread the knowledge. That is why I have published books and done exhibitions in the local languages. I have even published a series of stamps with the Antarctic theme, or even digitalize the types of mollusks in the Natural History Museum for 7 years. That was hard work. Right now, I am diverging. I have two incipient spin-off research lines. One of them is we're looking for new bioactive chemical compounds found in sea slugs, in the mollusks I am working with. And I'm working with that with a group of Chilean chemists, trying to find substances that could be useful for curing illnesses. The other research line is about bioluminescence, biofluorescence actually. We have found that many marine organisms have pigments that illuminate fluorescent when they are illuminated with ultraviolet light. And we think that this is a mechanism of species recognition. I'm working with this with several professors at the UP, a specialist from the CNRS, and also a group of colleagues from the Aquarium of Paris. Actually, you can see here, they have even, from this collaboration, they can even, they have done an exhibit showing the animals we're going to work with. It's amazing. To finish this talk, I would like to express one of my deepest dreams, and is teaching my students. I'm a vocational professor, to teach my students about biodiversity and tropical biology in the field. And for that, we have found a potential partner, Mero Foundation in Indonesia. And you can see in this image some of the photos that I took this summer trying to solidify the collaboration. I think that there's only a way to learn about biodiversity, and it's in the field. As a final reflection, as you all have been here in COVID times, I would like to think what would have happened if we would have valuable information about the different strains of COVID at the beginning of this modern plague. How many millions of people will have been saved? Well, there's a need to invest in learning about biodiversity. Thank you very much.